And we move on in view of time to the next speaker, who is Felipe Vilella from the Economics Foundation in Valencia. And he has a great track record already in working on human pregnancy and the endometrium. And that's also where the subject of the talk will be about. So Vilella, it's up to you.
Well, hello, my name is Felipe Vilella, and I would like to thank to the Human Cell Atlas to give us the opportunity to present our results in this amazing meeting. The title of my talk is Single-Cell RNA Expression of SARS-CoV-2 Cell Entry Factors in Human Endometrium During Preconception. In our laboratory, we are focused on working in the maternal contribution in the moment of the preimplantation, focusing on the endometrial microbiota, on the maternal embryonic crosstalk between the mother and the embryo, on the endometrial receptivity, and on the decidualization process. Today, I will focus in the potential entry of SARS-CoV-2 in the human uterus during preconception, and why, which is the relevance of analyzing this SARS-CoV-2 in the endometrium. This paper, published by Zheng et al. In JAMA Pediatrics early this year, showed that 33 newborns from mothers with COVID-19 pneumonia, 3 of the newborns have the symptoms of the COVID-19. And this indicates the possibility of existing a vertical transmission. This other paper, generated in New York by the Sutton Lab, published in New England Journal of Medicine, showed 215 obstetric patients presenting for delivery that the 84%, close to 85%, are negative for SARS-CoV-2, 13.5 are positive but asymptomatic for the virus, and close to 2% are symptomatic for the virus. This is pretty important to understand if this possible vertical transmission can exist. Let me introduce how the coronavirus infect the human cells. This is pretty important. Cell host tropism and receptor recognition by the viral spike S protein. This is the first step. And the second step is the priming of the S glycoprotein by the host proteases, activating the fusion of the virus onto the membrane, the virus entry into the cell, and coating the viral RNA into the cytoplasm. They perform the replication, transcription, and translation of the viral genome, and assembly of the viral genome inside of the nucleocapsid and envelope. Finally, they release the new virions from the infected cell. If we focus on the SARS-CoV-2, the host receptor is the angiotensin converting enzyme 2, called AC2. And the priming of the S proteins by the host proteases are the transmembrane serine protease 2, TMPRSS2, and the cysteine protease cathepsin B, and the same cathepsin L. These are the important proteins that are working with the infection machinery for this virus. The group of Lee, just published in the beginning of this year, mid of this year, in PLOS-ONE, using single-cell transcriptomic study, that the receptor AC2 is expressed on the maternal-fetal interface and the fetal organs. To perform this single-cell transcriptome, this was based on the single-cell RNA sequencing from 2 previous studies, the Bento Tormo paper and the published paper. They identified the expression of AC2 in bilious cytotrophoblast, in decidualized stroma, and perivascular cells of the decidua. They observed also a really tiny amount of expression of TMPRSS2, and looking for the expression of both in the same cells, there are just a really tiny amount of cells. If we focus on the AC2, they observed that the majority of the expression is in decidualized stroma. And for the TMPRSS2, the maximum amount of expression is in the epithelial glandular cells. We just published our paper them showing the deconvolution of the full endometrium at single cell level across the menstrual cycle. And we use our data to generate the expression of the SARS-CoV-2 infection machinery in the endometrium on women using this single cell level approximation. We work with 29 patients that 19 of these 29 patients were analyzed using a fluidigene C1 system using microwells in single-cell RNA-seq, analyzing a total of 2,148 cells, and then focusing on the window of implantation and the residualization process. We was working with 10 patients, analyzing more than 70,000 cells, but for that, we was using the platform Chromium 10X using the droplet system that give us more profundity and give us more possibility to analyze cells. Also, to compare both results, we sequenced two different sets of samples from the same patients. If we focus in the fluidigene C1 microfluidics, and if we focus on the epithelial, glandular, and luminal, and also in stromal cells, we analyze the expression of the different factors of the SARS-CoV-2 cell entry. We observe that AC2 is not expressed, or really tiny amount of cells are expressing AC2 in epithelial cells and neither on stromal cells. If we focus on TMPRSS2, we observe a tiny amount of more expression on epithelial cells and really a small amount of expression in stromal fibroblasts. The situation is completely different when we are looking at cathepsin B and cathepsin L. The expression of cathepsin B and cathepsin L in epithelial cells is pretty high, and also in stromal fibroblasts. Analyzing that more in detail, we observe that the AC2 is nearly not expressed across the full menstrual cycle, TMPRSS2 just a bit in the early and proliferative stage, but really tiny amount in the other phases of the menstrual cycle. But cathepsin B is highly expressed in the majority of the moments of the menstrual cycle, in the majority of the cells, epithelial, luminal, epithelial, glandular, and stromal, and just observe a less expression during the process, during the moment of early secretory. Cathepsin L is less expressed than cathepsin B. If we are looking for the full machinery, the expression of both of the factors, if we concentrate in AC2 and TMPRSS2, that is the one that is defined, that's the main machinery for the cell entry for the SARS-CoV-2, we just found 4 cells that are expressed across the menstrual cycle, and the amount of cells that express both, the co-express both of the factors is really low. We focus in AC2 and cathepsin B, we observe similar results, and same from cathepsin L. This is just because the expression of AC2 is really low in the epithelial cells and stromal fibroblasts. If we are more in deep going during the moment of the window of implantation and as well during the moment of desitualization and focusing on stromal and epithelial cells using these Chromium 10X droplets, analyzing more than 70,000 cells, we see similar results in the case of AC2 and TMPRSS2. The expression of AC2 is really low in epithelial, practically is inexistable in stromal, and it's a bit more expressing of TMPRSS2 in epithelial cells, and practically there is no expression in stromal cells. And we observe similar results for cathepsin B and cathepsin L, more expression of cathepsin B in both epithelial and stromal cells, and less expression, but pretty high compared with the other factors of cathepsin L in epithelial and stromal cells. If we are more in detail, practically no expression of AC2 across this moment of the menstrual cycle, neither of the TMPRSS2, high expression of cathepsin B and less expression of cathepsin L, but still pretty high in some specific moments of the menstrual cycle, and focusing more this cathepsin L in stromal fibroblasts. If we focus in the co-expression of the machinery of cell entry of SARS-CoV-2, AC2 and TMPRSS2, we observe really tiny amount of cells. We see a bigger increase of AC2 and cathepsin B expression, co-expression. We have around 330 cells in total, but if we compare with the 72 1,000 cells analyzed, this represents less than 0.01% of the cells that are expressing both factors, and less expression of AC2 and cathepsin L. With that, our conclusions will be that AC2 presents measurably expression in all endometrial cell types during the preconception period. High expression of cathepsin B and cathepsin L may imply that some part of the SARS-CoV-2 cell entry machinery is active in the endometrium at this time, but is not the machinery that is already described in the bibliography that is implicated in this SARS-CoV-2 entry. And our findings suggest a low efficiency of SARS-CoV-2 viral infection in the human endometrium before preconception. If we analyze the results of our colleagues, Lee et al., published in PLOS ONE during pregnancy, they conclude that the early human placenta shows high expression of AC2 in 4 main cell types. AC2 and TMPRSS2 expression in maternal-fetal interface may increase along with the trimester of pregnancy, and despite the distribution of AC2 and TMPRSS2 in the C1 placenta, the existence of vertical transmission of placenta dysfunction caused by SARS-CoV-2 infection is just speculative at this moment. Well, I would like to thank the organization again to give me the opportunity, and I'm happy to get questions. Thank you so much.
Okay, Philippe, thank you for this very interesting and very detailed talk. There's some questions from the audience, but I have a question myself that I'd like to ask first. That's, I think, my prerequisite as a chair. In the airway epithelial system that Kirsten just presented, it's well established that ACE2 is an interferon-induced gene. So under certain circumstances, expression is being induced. Do you have any idea whether the same holds true for the endometrium?
No, we don't. We didn't check it out.
So, because of course it's very interesting to consider if 10% of the children born from mothers with COVID-19 are positive, how is the transmission then achieved? Do you think it is through the placenta, or?
It could be through the placenta. It's the more logical situation. But our analysis is more focused in the preconception moment, in the beginning of the preconception, just in the moment of the window of implantation. But for the child that are born for the vertical transmission, it looks like should be by placenta, but it's not already demonstrated.
Exactly. Okay, thanks. There's one question from the audience where the question is, what about male fertility, since ACE2 is highly expressed in sperm? Have you investigated this, or are you planning to?
No, we didn't. We just focus on the female reproductive tract.
Mm-hmm.
We are not working on males.
So then another question could be, because the relevance for the children, of course, is very, it's very important to understand how the virus could affect the children and their health. So are you aware of any studies that have looked in these children, follow them up once they are born from COVID-19 positive mother and then after vertical transmission?
I didn't see any study that are focusing on that, because everything is really new and we need to this follow up, for the next few years. we'll have results in the next few years. But actually, I didn't see any study. Maybe with other viruses or other SARS virus, it's possible, but I didn't see anyone.
Mm-hmm. Okay, well, thanks again for your very clear presentation. If there's no further questions from the audience,